And our first team today is a quick start that was implemented in Bangladesh, and the title of the project is Genome Sequencing and Development of SNP Markers from Rohu in Bangladesh. And I will turn it over to, I believe, Professor Khan, who will start us off.
Yeah, can you hear me? Hello? Yeah. Respected researchers, good morning, good afternoon, and good evening to all. Our research mainly, this is the part of genome sequence research project, a quick start project, and the mainly this is the previous slide, our previous slide. Our title was Economics of Rohu-based carb polyculture in Bangladesh, Yield Gap, Youth Involvement, and Nutritional Perspective. Please next. And as you know, the fish farmer usually expects the higher yield and return, but due to several factors, they cannot get expected yield. The farmers' efficiency and financial resource availability may be the factor that can result in lower productivity. Thus, there is difference between the farm level yield and lab-based yield, and that result in yield gap. So besides, there is a productivity differences between the Rohu-based carb polyculture and monoculture-based system, which is mainly Pangasius and Tilapia monoculture. Therefore, this Pangasius and Tilapia monoculture, their productivity is 3 to 4 times higher than the polyculture system. Therefore, farmers are not interested to culture carb-based polyculture, even though they have higher market price and consumer acceptance. There is a tradition in Bangladesh that when a new son-in-law visits to his father-in-law's house, he has been served by a head of big Rohu fish in his meal as a prestigious value. So this would be evidence the appreciation and the popularity of Rohu fish in Bangladesh. So based on our research, our objective was next. Please measure the profitability and profit sensitivity of Rohu-based carb polyculture, assess the efficiency, yield gap, and youth engagement in polyculture, and analyze the species preferences of carb polyculture and nutritional status of Rohu. So please next. To fulfill the objective, the study carried out in the 5 districts of Bangladesh based on the Rohu production, and we have selected 183 samples. And we have used several econometrics method. I am not going in details to fulfill the objective. Please next. So this is the research findings. Time is very limited. That's why I'm going very fast. So from this graph, we can obviously remark that Rohu almost contribute half of the total production. That means 43% of total polyculture production. However, it is estimated that per hectare productivity was only 5.6 ton per hectare, and it is 3 to 5 times lower than the Pangasius and Tilapia productivity. Despite that, Rohu-based carb polyculture also profitable enterprise, which is we can ensure by to see this year, that is benefit cost ratio was found 1.38, implies that if farmers invest $1, they can earn $1.38 from $1 investment. And it was profitable because it requires low feeding and management cost. Next, please. Next slide. Yeah. This study found that only feed cost consists more than 60% of total variable cost. Now, this previous slide, please. To generate some sustainable policy, we try to exhibit the effect of alternation in feed cost and fish price on financial benefit of the fish farmer, where 6 scenarios were evaluated with the business as usual situation. And from this table, we can see that we have 6 scenarios with business as usual. And results shows that if we can reduce the feed price by 10%, with the keeping the feed quantity unimpaired, the BCR raised to 1.42. So it would be the best solution for farmer. From this result, it can be concluded that the reduction of feed price or increase in output price may be an effective policy for sustainable Ruhu-based carb polyculture. Please, next slide. We estimated the technical efficiency level of the farmers, and we found that the average or mean technical efficiency was found 69% of the sample farmer, which implies that farmers could below the frontier level, and they could increase their output by 31% with the same level of technology. And we also found that this production was positively influenced by fingerling, feed, fertilizer, lime, water cleaning cost, and the vitamins used. And we also found the farmers' experience training and extension service positively affect farmers' efficiency. Next, please. This slide shows that this figure shows that the highest recorded yield gap we found was 5.78 ton per hectare, and potential yield gap was lowest, this 1.76 ton per hectare. We estimated 3 types of yield gap: the higher recorded yield gap, experimental yield gap, and potential yield gap. And besides, we observed a positive relationship. This right-hand side slide, the 3 types of yield gap, and we found a positive relationship between inefficiency and yield gap. This implies the efficiency of the fish farmers could boost up by reducing the yield gap. Please, next. Next, please. So we try to find out what is the causes of, previous one, please. Causes of yield gap, and we identify the abiotic and biotic factor for yield loss. And result clearly, this shows that abiotic factor are more responsible for losing yield than a biotic factor. However, the total loss due to abiotic and biotic factor in the affected area was 14% of gross yield. Among the abiotic constraint, water quantity was found the prime factor, and this water quantity was comprising of water shortage and the flood in the study area. Next slide, please. These 2 figures shows that the result of the study discovered this is the youth engagement in the aquaculture of Bangladesh. The 32% of the farmer study were in age group between 18 to 35 years that was considered as youth. And however, productivity and efficiency of farmer increases with age. The right-hand side, this figure shows that reaches some mid-age, mid-peak, and then decrease with further age increase. This mid-age of somewhere 40 to 45 years, that was the suitable age for increase the productivity and efficiency of the farmer. So next slide, please. This 3 figure shows, we try to show the relationship between age and yield gap, and we can see that the yield gap decreases up to certain level of age, that is near to 45 years, then the yield gap increases with age. So young farmers have less yield gap in the production process. That means young farmers are more efficient and productive. Next, please. This is the preference of species preference, because this was monoculture system. We found that most of the selected farmers prefer Ruhu as their main species in culture, followed by silver carp, katla, mrigel, and mirror carp. However, higher demand, better FCR, better flavor were the major reason behind the choices. We found from this 2-bit model that we have used for our analysis. Next slide, please. This is the nutritional status or consumption of fish. So in the study, a farmer consumed a mass amount of fish due to availability. It was an important finding that average daily consumption of raw edible fish from own culture was 77 grams per person. The graph revealed that each person consumed 41 grams of raw edible roe fish per day, which contributed 57% to the total daily fish consumption from own culture. However, roe contributed about 14% to the total daily protein requirements for an adult of the selected farmers, and it alone contributed 1.65% to total daily energy requirements of the farmers. So please, next. From this result, what is the lesson learned from this study? So roe-based polyculture was profitable, though farmer could get tremendous profit. It was possible to increase fish price and decrement of fish price. The efficiency level of farmers was below the frontier line, and they could increase 31% production, and there was considerable amount of yield gap exist in the current production practices. Young farmers are more efficient and productive. Fish farmers prefer to culture roe for higher demand in the market and better FCR. Fish farmers consumed significant amount of roe fish than the other species due to the better taste and flavor, and roe contributed remarkably to their daily protein requirements. Next, please. This is the last slide. Recommendation and further research. So the price of feed should be kept as reasonable as possible to compensate the farmers and enhance the quality of feed. More research on feed should be taken place. Farmers should follow the scientific management practices to reduce the inefficiency and yield gap. Training and research on technical knowledge and farm management systems should be strengthened. Disease has effect on losses of yield. Research on fish disease control and genetic improvement need to be ensured. According to respondents of the survey, the genetic quality of the current hatchery stock of the roe has deteriorated significantly, and an improved stock of roe is needed to boost the roe production, and that could be achieved through marker-assisted research selection. Also, I would like to add 2 more recommendations here. The research on fish quality and safety is important, what we observe in our field survey, and research on how youth and can participate in the aquaculture farming system need to be investigated. Thank you for patient hearing. This is all about from my part. Thank you.
Okay, hello. My name is Dan Peterson. I'm going to talk a little bit about roe genomics, and specifically sequencing of the roe genome and some other experiments we did for looking for variation in the genome of roe. There was lots of people involved in this project. Mississippi State University, Bangladesh Agricultural University, World Fish were all involved in this. We kind of Mississippi State here today. Next slide. I'm going to keep this very, very short. Essentially, the genome of an animal or of an organism is essentially the genetic blueprints of that organism. And from those blueprints, if you will, to have those in hand gives you a lot of leverage on how you can approach improving the fish, how you can approach understanding them, and how they interact with their environment. So one of the first things that we did is what had been reported previously for Rohu was that its genome size was 1,950 million bases long, which seemed pretty high to us. It's about twice the size that had been observed for other carp that were closely related. One of the first things we did is actually looked at, we actually determined the genome size using probably the most standard means of doing it, called flow cytometry. And the previous value of 1950 had been reported using something called folding densitometry, which is a great technique, but it's pretty hard. And what we found doing the flow cytometry was the genome size was actually about 968 million base pairs. So roughly half the size of what was previously reported. And as I'll go and show you, this is kind of held up under our study. That was a first real discovery on the genome. The next thing we did is we sequenced the genome. This is not an easy task. I won't go into the details, just to say it's essentially, if you could pull out the DNA molecules out of the nucleus and line them up like this and sequence them from one end to the other, that would be fantastic. So essentially, we end up extracting all these little pieces. Get millions and millions of pieces of DNA, and we have to try to put them together and the order that they are actually inside the malignant cell. So it's a big puzzle, and we utilize supercomputers and high-throughput sequencing and new sequencing techniques to do this. We assembled a version of the genome based upon our sequencing techniques, and what we got was about 940 million base pairs that assembled into what looks like a genome. So that's roughly 90% coverage of the genome as we determine the size, which agrees perfectly with the size determination and there's a value called N50. This is kind of roughly the length that these 50% of the pieces that we had were greater than found in sequences that were greater than 10.5 million bases long. So we had some very large pieces, which was very good. Next slide. One of the things that was interesting is as we were working on our genome, somebody else published the same genome that they had put together. And this happens a lot in this field. Generally, in my experience in sequencing genomes, if you've got a good idea, someone else probably has that same idea. So another group published genome of Rohu. Our assembly came out being 5 times more complete than theirs, and without certain duplications that were a problem in theirs based on the way that they assembled it. So right now we're working on a paper to describe the genome, and this is one of those things that has kind of had to wait until the end of the semester, because this has been a semester from hell for me in terms of trying to get everything done with COVID and so on. But we are working on this. What is the value of having this genome sequence? Well, one of the real value points is that it serves as a standard against which we can gauge the diversity of the species. And so we essentially get a sequence, a really high quality sequence for one individual, and then we can do some really quick sequencing and compare other individuals to that. We can say, oh, okay, here's a difference here, here's a difference here. And we can start looking at the differences, but also looking at differences that occur in the phenotype and the very physical characteristics of the fish. This kind of genome, and it's a very high quality sequence I may add, our genome sequence will facilitate advancing of molecular breeding techniques. And this is something that we've been talking with our colleagues at Bangladesh Agricultural University and World Fish, is actually utilizing and leveraging this genome sequence to improve the fish, improve the rate at which they grow, improve other characteristics, disease resistance, and so on. And so we've done this for Rohu. Next slide. We did another little study, which was based on fin clips that were taken from different fish, different rohu. And unfortunately, there wasn't a lot of phenotypic data. There wasn't a lot of data on the characteristics of the fish. But one of the things that we could do was look at the source of the fish. And the fish were taken from 3 different rivers: the Halda, the Jamuna, and the Padma River, which is shown over here to the right on this peak. With Halda being red, Jamuna being green, and Padma being blue. And this is just looking at the diversity graphed out on this. I don't even understand the graph that well, except that if you look at it, these red dots are the Halda River, and then the blue and the green dots are the other 2 rivers. And you see the other 2 rivers, the fish are genetically more similar. They're all kind of interspersed. And then if you look at the Halda River, everything's kind of over here, which means more genetic diversity, or they're more diverse than these other groups. And this is not, as we started talking with our colleagues in Bangladesh, we started learning more about these river systems. And truly, it's the Padma and the Jamuna rivers run into each other. I don't remember which one's the river that runs into the other one, but it's kind of like the, a branch of the Mississippi River is the Missouri River or something like that. It runs into the Mississippi. They're all continuous. So it's not surprising to see this. And actually, this is a river that's separated from those other rivers. And that does explain some of this variation. But this is just a little preliminary study, but we'd like to get the opportunity to look at more characteristics of the fish, including physical characteristics of the fish and so on. And that's it. It's like, yeah, that's awesome.
Okay.
Excuse me, Dan.
Yeah.
It's a bit difficult for us to hear you. Do you have a headset or?
You're having difficulty hearing me?
Yes, sir. Not
Okay.
really hearing, but the quality.
I'm not sure. I don't have a headset with me. Do you need anything? Would you like me to say anything over, or I'm kind of done now at this point?
Oh. Maybe if anybody could chime in, if you wanted clarification or if you needed him to repeat, please let him know.
Thank you, Dan. And thank you, Professor Khan. So, right, so this project had 2 components, and Professor Khan led the survey portion of the project, and then the sequencing and SNP investigation of the rohu was the second part. And due to time, we'll need to limit this discussion to only 1 or 2 questions or comments. Are there any questions from the group?
Hi, Mark. I have a question for Peterson. Just because we did have trouble with the audio, Peterson, if you could just please summarize maybe in a few sentences or bullet points, really what were the findings from this activity. I'm sorry to have you repeat it, but just so we all get the major findings. Thank you. Over.
So I didn't even hear what that was. What was that, Mark?
Yeah. Yes, Dan, could you summarize quickly the final results from your project?
Right, and I'll try to speak loudly. I hope this helps. Our part of the experiment, our final results is we put together a very high quality, one of the best genomes for fish, in particular the rohu. And that took quite a bit of time and effort. But we put together this fish genome. We are now looking at diversity within rohu. By comparing the standard reference genome with other genomes of rohu fish. The other thing we really are looking to do is work with our partners at World Fish and Bangladesh Agricultural University to start improving the fish based upon molecular breeding techniques. And in terms of other discoveries, we discovered that the genome of the rohu is half the size that originally was reported in the literature. And our genome sequencing supports this as well as our independent characterization of the genome. And I guess that's about it.
Thank you, Dan. Okay. Any other final? Yes.
Mark?
Yes, Mike.
Just to say, Dan, really interested to see the results. I know we're very keen to explore application of some of the results to the genetic improvement program with rohu and other carps in Bangladesh. So I'm sure we'll have further. I look forward to further opportunities there, and it's great just to catch up and see the results presented like this. So thanks very much for the cooperation. Really interesting work, and I enjoyed the previous presentation as well on youth and yield gaps. I thought that was fascinating, and I was joking with Rahana that we're too old to be efficient rohu farmers. Fascinating research, and I really enjoyed both presentations. So thank you. Over and out from me for now. Anyway.
Thank you.
Thank you, Mike.
Mark,
Yes.
Melba had a couple of questions in the chat room. I can repeat them if you like, or you can.
Oh, I see.
them. Oh, I'm sorry.
Could you go ahead and read them, Chauncey?
Okay, Melba, if you would go ahead and speak yours, and then Dr. Alam, he put his in the chat.
Okay. Hi, everyone. Can you hear me?
Yes.
Yes.
Yeah, okay. Thank you very much for the presentation. I have two or three quick questions on the methodology. I wonder what criteria were used in selecting the 183, I think they were survey respondents. And I just got this from the quick startup summary. You mentioned that these were commercial farms. So could you elaborate a little bit on what you mean by commercial? And then the third question is whether, I was wondering whether in this collaboration you have engaged the competent authority in Bangladesh, because I think that although this is very, very much an academic research, I think at the end of the implementation of the research, it's important to make a follow-up on how these research findings can be practically implemented at the ground level, and whether the research findings can be converted into, for example, policy changes as far as the government is concerned. So it's important to get also the buy-in from the competent authority. Thank you very much. Over to you, Mark or Chauncey.
Thank you, Melba. Time for a quick response from the team before we move on.
Yeah, thank you. I am Khan from Bangladesh Agriculture University. So this question was about how we have selected 183 commercial farmer from the field. So this was mainly we, of course, we have applied the statistical method for sample selection, but first, this was three-stage stratified random sampling process. First, we selected which area is more productive for Rohu-based polyculture, and we selected 5 districts on the basis of Department of Fisheries Statistics. This was mainly purposefully selected. Then we randomly selected the fish farmer. We have collected the Rohu fish farmer list from the Upazila Fisheries Officer. Upazila, this is mainly sub-district of Bangladesh. And afterwards, we selected randomly this farmer. So I think we are not biased on the selection process. So in this way, we selected 183 Rohu-based polyculture farmers from this 5 districts of Bangladesh. And also, you are talking, your question was how we can disseminate the result from policy to the farmer and policy or policy changes. As you know, we are academician, so we have found some result, and we are trying to publish this paper. Also, Planning Commission was involved with this with us, and Bangladesh Fisheries Research Institute, this BFRI, also was involved with us, and they are mainly their extension partner. They disseminated the result among the farmer. They have many training with the farmers, so they will disseminate our result, this BFRI and the Planning Commission, and as academician, we will publish our paper within a few months.
Thank you, Professor Khan.
Yeah.
Dr. Mark, can I just make a small comment?
Okay.
Actually, when we did the survey, we met all the district fishery officers